Okay, hello everybody. Welcome to our next invited speech. I'm going to introduce Dr. Ziad Hafed, which basically by training as an electrical engineer, getting his, as far as I understand, BS, MS, and PhD from McGill University. He moved to Salk Institute for his postdoc and worked with Richard Krausless, which I had the honor of knowing when I was in NIH. And actually, I think one of the postdocs of Richard, Amar Badaghi, is now working with Ziad. We were at the same time in LSR together. Ziad is one of the probably best experts in Superior Curriculus, mechanisms of Sacada, Microscopes, and he's one of the highly cited, respected researchers in this regard. And he has received a number of prestigious rewards, and produced great papers. Some of the recent rewards is the British Association Medal for Great Distinction, and NCERC, and the Sloan Schwartz Fellowship for Neuroscience. And from 2010, Ziad is in Tübingen, and he's leading a research group. And again, so he's telling me that there are a bunch of papers that is very close to his heart, including a bunch of papers from 2013, Hafed, Chen et al. 2015, Tyan et al. 2016, Hafed and Chen 2016, which people can search and read about. And I guess today Ziad will talk about some of the science related to these. The title of the talk, as you can see, is Unraveling the Neural Mechanisms of Perceptual Suppression. So we are honored to welcome him, and we're looking forward to hearing his talk.
Okay, thank you very much. I hope everyone can hear me and see my slides well. So first of all, I'd like to thank you for the opportunity and the invitation. It's an honor, and I wish it was for real. And I have actually tried to attend as many of the talks as possible, and they were all very informative and very helpful, and a learning experience. And that's always a good thing. So my lab is interested in the sense of vision, the visual modality, and we take an active perspective in this regard. So we believe that to study vision, you essentially end up having to study eye movements as well. And this is because eye movements are an integral component of vision. The very least reason being that you need to basically realign your fovea with different image regions that might be of interest to you in order to be able to understand the entire visual scene. So this scene is incidentally right outside of our city of Tübingen. It's a much smaller city than Tehran, I would imagine. I've never seen Tehran, but I would bet that it's a much smaller city than Tehran. And so we need eye movements at least to scan the environment. And as a result, we do vision by doing eye movements. And we do this, at least with saccades, very frequently, more often than the heart beats, basically, at rest. Now, this is useful because we allocate the fovea to objects of interest, but it also creates a new dilemma for the visual system, because if you were to look at this same scene from the perspective of the retina, then you would find that the retina is basically experiencing a jittery movie continuously all the time. It's always shifting, it's always jumping. And our subjective perceptual experience is one of stability. In fact, if we continued looking at this for a while, we might suffer from motion sickness and so on. And therefore, this was just in case the movie didn't work, but vision is essentially a series of fixations, indicated here as circles, interspersed by these sudden jumps, indicated here as arrows, which are saccadic eye movements, to look at other regions of the image. And so you create a spatiotemporal movie on the retina, even if the image is completely stable. And as a result, the brain, when it does vision, is continuously engaged in this perpetual loop. So on the one hand, you have visual perception and processing and perception, getting the input, and then at any one moment in time deciding where it should look next, or what it should do with its eye or its head or its body. And then the flip side of it is that every time you move your body, then you change the sensory input and therefore need to process what is being changed because of your own self-movement. And so we are driven in the long term by the question of what are the neural mechanisms that link perception and action. And we study this by performing behavioral experiments in human subjects and also in non-human primates, rhesus macaque monkeys. And we try to do essentially the very same behavioral experiments in both species because we're interested in the human condition ultimately. But the non-human primates now give us the opportunity to go invasive and do neurophysiological experiments. And then we also try our best to complement the experimental work with some modeling stuff that allows us to test specific hypotheses about our results and maybe even generate newer hypotheses. So for today's talk, I would like to start by focusing on the superior colliculus, which is the midbrain structure indicated here on the brain of the rhesus macaque. So we heard about the colliculus yesterday from Mike Shadlin. Our interest in it is from a sensory perspective. So the colliculus has several layers of neurons with two functional layers, basically a superficial layer that contains purely visual neurons, and some of the input to this layer is actually directly from the retina, and the rest is from early cortical visual areas. And then the deeper layers which move the body, move the eyes and the body, also has visual responses in these so-called visual motor neurons. So these are sort of multi-dimensional neurons. They don't just do saccades, they do vision as well. So we think that the superior colliculus is a perfect candidate circuit for studying active vision because it basically has all the elements. It has eye movement generation, it has the decision making that Shadlin alluded to yesterday, and it has visual sensory processing, in fact very early visual sensory processing because it can receive direct retinal inputs. And so for the talk today, I would like to now take this general question and focus it onto the question of saccadic eye movements and how perception is modulated around the time of saccadic eye movements. So we're interested in basically perisaccadic perception today. Okay? And again, the idea here is that whenever we move the saccade, the eye, the fovea with the saccade to inspect a new visual scene location, what we are doing to the retina is introducing a rapid image shift that should normally go perceptually unnoticed. And this perceptual cancellation has been studied for many, many years with a technique that is basically trying to aim at identifying or probing the instantaneous state of the visual system. So what you essentially do is you input something to the visual system as a probe to try to understand its instantaneous state. And what I mean by this is that if you imagine a scenario like this, where the vertical line here is the onset of one saccadic eye movement that you're about to make, okay? And then at different times, the x-axis would be time. At different times, you would present a very brief probe stimulus, like a white dot, for example, over a black background. And what you do is you try to measure the sensitivity of the visual system to this white dot, here written as perceptual detectability, as a function of time relative to the saccade. So the brief stimulus acts as if it's like an impulse to the visual system that captures the instantaneous sensitivity state of the visual system. This is analogous to stop motion photography in the early days of stop motion photography. And so what you do is you do this for different times around the saccade, and what you find among the phenomena that you find is that perception changes as a function of time relative to the onset of an eye movement. So if you present a stimulus a long time before saccade onset on the left, or a long time after saccade end on the right, then you perceive the stimulus veridically, you perceive it very well. However, if now I present the very same stimulus around the time of the saccade, then the sensitivity, the instantaneous state of sensitivity of the visual system is strongly impaired, and that's called perceptual saccadic suppression. And it gets so extreme that basically at the peak here, you literally do not see this probe stimulus, even though during fixation it's suprathreshold and it's very easy to see. So this is perceptual saccadic suppression. And if you were to now present a stimulus to a neuron, let's say in the superior colliculus, so time 0 here would be the time of stimulus onset, then if you present a stimulus to the neuron's receptive field without a saccade, you get a very brisk response in this red curve. And if you now present the very same stimulus to the same neuron, but in this case immediately after the end of the saccade, so all the image motion on the retina was already completed, then you get this reduced visual sensitivity. So this is a neural correlate of this idea of perceptual saccadic suppression. So as I said, this phenomenon is very robust. It's replicated in many labs, and it has been intrigued vision scientists for many decades. And along the way, there have been strong controversies. There have been strong fights. And among the fights are basically what is causing the phenomenon. The phenomenon is so robust, it's repeatable, but what explains it basically? So one possibility, one camp of people argues that this phenomenon on the left here is basically a phenomenon that's indicative of concepts that are very popular in several fields that are related to these concepts of efference copy or corollary discharge. The idea being that you have internally a motor command that is being generated. For example, the superior colliculus will generate a motor burst to move the eyes, just like we saw with Shadlin's talk yesterday. And so this motor command can then now inform visual areas that the eye is moving and therefore be a command to suppress sensitivity. So the command is sent to the eye muscles, but also sent to the visual areas to suppress sensitivity. On the other hand, you have people that argue, well, no, I can design a visual system that can actually suppress sensitivity as a result of image shifts or whatever without the need for knowledge of the motor command. And so then there was an either or, and there were big fights basically. So this is part of the controversy, and some of the another part of the controversies basically is what I would like to focus on today, which has to do with the properties of perceptual saccadic suppression. And I particularly clarify this with one very influential paper that was published by David Burr and Concetta Moroni and John Ross about, now it must be 26 years ago. And so this paper is describing a very robust property of perceptual saccadic suppression, which is that it exhibits certain selective properties. So the title here says Selective Suppression of the Magnocellular Visual Pathway During Saccades. And I'll explain this a little bit, but that's basically what the title is. So I'd like to explain this selectivity in more detail because I'll come back to it at the very end of the talk. So what did these authors find? So the authors essentially did psychophysics. So they did behavioral experiments. And I told you that you present a very brief flash at different times relative to the saccade. What the authors did in this study was replace the flash of like a white spot with basically a pattern. Like here I show a low spatial frequency pattern on the left or a high spatial frequency pattern on the right. So now they measure the perceptual sensitivity, not just whether there's a white flash or not, but whether there's a low spatial frequency flash or not versus a high spatial frequency flash or not. And so the x-axis is the type of flash that they presented around the time of the saccade, so this perisaccadic interval, and the y-axis is the sensitivity of the visual system. Okay? So because the visual system sees different frequencies with different efficiency, you need the baseline visibility. So the black dashed curve here is basically how well you see the flash without any saccades. And we know that there's a contrast sensitivity curve, right? So we know that low spatial frequencies can be seen well, depending on how much contrast you give to them, but high spatial frequencies get a bit harder to see, okay? So this is the baseline veridical sort of visibility of the flashes without any saccades, okay? Now, if you do the saccadic suppression experiment, you present these same flashes in this peri-saccadic interval. And what you find, or what these authors found in this influential paper, is that the low spatial frequencies, here the lowest 2 or 3 spatial frequencies, have very strong suppression. So something that you see perfectly without a saccade becomes almost impossible to see with a saccade. But then if you go to this third spatial frequency, if you increase the spatial frequency, you actually get better. So the suppression effect is selective. You only suppress low spatial frequencies and you do not suppress high spatial frequencies. That's exactly what these authors found. It's a very robust phenomenon. It's very repeatable and highly influential. So why did I say that it's controversial? Well, for one, the title is very neurophysiological, so it makes a very clear prediction about neurophysiology, but it was a purely behavioral experiment. And people have searched for a neurophysiological correlate of this behavioral result in these 25 years that have passed, and there was no convincing neural substrate for it. So that's why it's sort of begging to be looked at, right? It's not satisfactory to sit with such a robust phenomenon for 25 years without convincing neural substrate. So we started out by looking at it, and we had reason to look in the superior colliculus, because I told you earlier that we have a neural correlate of suppression in the superior colliculus anyway. So now let's look at this selective property of perceptual saccadic suppression. So we went in and tried to look for a neural correlate of this very robust phenomenon. Okay, so a neural locus for this perceptually relevant saccadic suppression, and this was done by a former student, Chen, who is now a postdoc in Japan. So he figures in a lot of my favorite papers, as you heard in the beginning. So what did we do along with Chen? So we did the experiment by exploiting microsaccades, because these are essentially small saccades. So if we ask you or a monkey to fixate on a spot here, you will basically be generating small saccades. And these, from the perspective of this experiment, are interesting because they don't move the retinal image by much. So we can have a visual receptive field of a neuron that sees this flash that comes into it, okay, without having to worry about, where is the flash in the receptive field and do we have enough display room in the lab to present the flash or not. So from a perspective of a lab with a limited display size, this is practical because the microsaccade is small and therefore you can run the experiment in the lab with a small display. So what we did was we presented a flash in the receptive fields of superior colliculus neurons. We know that there will be suppression from past work. And so now we'll do this immediately at the end of a microsaccade so that there's no retinal image jitter, basically. The image jitter has finished already, okay? And so now we run the experiment with different patterns. Instead of just a white flash, we present different patterns to the receptive field of the neuron. So either a low spatial frequency, 0.5 cycles per degree of visual angle, or up to, on the right side, 11 cycles per degree of visual angle. Okay, so what do we find? So I'll show you an example. In this case, it's a visual motor neuron, so it's from the deeper layers, but we only look at visual responses. This is a fixation task. The monkey does nothing with the stimulus, okay? So again, time 0 is the onset of the stimulus, and in this case, we present a low spatial frequency stimulus, and we find a robust response. And as you might expect, we also found saccadic suppression. So if the very same stimulus is presented now immediately after the end of a microsaccade, you get very strong suppression. And now what I'll do is I'll cycle through different spatial frequencies to go higher and higher, and we compare basically the red and the gray curves, okay? So for low spatial frequencies, there's a big difference. If we go to a higher spatial frequency, we still get suppression, but the difference between red and gray is reduced. And then for an even higher spatial frequency, we get an even smaller suppression, okay? And then by 4 cycles per degree, basically the baseline response and the suppression response are essentially identical, and similarly for 11 cycles per degree. So now we found selective suppression, right? In the sense that if you now plot on the y-axis a measure of visual sensitivity of these neurons, and on the x-axis the spatial frequency of the flash that you present around the time of the saccade, you see that the low spatial frequencies have reduced sensitivity, so there is stronger saccadic suppression, and the high spatial frequencies have weaker saccadic suppression. They're not affected by the saccade as much as the low spatial frequencies. And we also, in the same animals, designed a behavioral experiment to look for a perceptual cost, if you will, of the saccade. And so now on the y-axis in the right panel, is a measure of the perceptual cost, so a reaction time cost. So if there is more reaction time cost, then this is a bigger impairment. And so you see, again, consistent with this influential paper from 1994, that the low spatial frequencies have a big impairment and the high spatial frequencies have a weak impairment, exactly like the collicular neurons. And there's a very clear correlation between neurons and behavior in the superior colliculus. So we were very happy because we now found a neural substrate for this selective property of perceptual saccadic suppression. Okay, so this was very interesting. However, as is the case with science in general, the more you learn, the more you discover mysteries, because basically it was a victory, but at the very same time it was a kind of defeat. Okay, and the reason that it was a kind of defeat is that now, if we looked in the very same structure in the superior colliculus, not just at these visual motor neurons in the deeper layers, but at the visual neurons in the superficial layers, we found something surprisingly very different. Okay, so I'll show you an example, visual neuron, and here for low spatial frequencies you get suppression, but you already see that it's much weaker suppression. It's almost non-present. The visual response is almost unaffected. And then if you go to higher spatial frequencies, you get the same amount of suppression. So basically there is no selective suppression, it's just constant suppression. So in the same circuit, the superior colliculus, some types of neurons will not be selective for low spatial frequencies. So they violate this robust perceptual phenomenon from this influential paper of Burr and colleagues. So this was very surprising because the intuition from the literature on the superior colliculus is that the deeper layers, here indicated as SGI, basically carry the information about the motor command up to the visual layers, here described as SGS, and then up to the cortex via these upwards going pathways. So this is sort of the efference copy or the corollary discharge hypothesis. And so what we find is very different because we find strong visual suppression in the deeper layers, almost no suppression in the visual layers. So this pathway might not be it, right? So we question now whether the motor commands from the deeper layers really do suppress the superficial layers and then ultimately the visual cortical areas. So this is sort of back to square one, essentially. And the reason that it's also kind of a defeat is that now, okay, we did find a neural locus for this influential phenomenon that is so robust, but at the same time we found that there are neurons in the same structure that will completely violate this phenomenon. Okay, so what gives? We're almost back to square one. So this made us completely reset. So we decided that, okay, let's step back completely from this whole history of saccadic suppression and so on, and let's clean our slate. Don't take sides. Don't assume motor commands. Don't assume visual commands or visual mechanisms, and let's just go back to very first principles. And by very first principles, I mean, what is it that happens in the retina and in the visual system when you make a saccade? Okay, so if you remember, I said that when we make a saccade, we're essentially creating a movie on the retina. So let's go back to the very first visual processing stage in the visual system. Let's go back to the retina and think of saccades from the perspective of the retina. And saccades from the perspective of the retina are actually a strong stimulus. It's a movie. It's a sudden shift in the natural image. So rather than taking sides, we say, okay, we won't take any sides now, but from first principles, a saccade is a stimulus to the retina. So let's try by asking, to the extent that there are visual mechanisms for perceptual saccadic suppression, what is their first neural locus in the visual system? And as I said, we go to the very first area, the retina, okay? And if we do find something relevant for perceptual saccadic suppression in the retina, what is the contribution to the perceptual phenomenon? So perception is really the output of the whole brain. So we're going to go to the retina and try the very first input to the visual system and try to relate its properties to the output of the entire brain, which is perception. Okay. So we exploited the fact that our next door lab has retinal electrophysiology capabilities. And so we recruited two outstanding students. And this was really a beautiful collaboration, right? So Matthias Baumann on the one side would do perception in humans and monkeys. And then Saad Idris on the other side would take the predictions that Matthias makes. He would tell Saad, I predict this from the retina, let's test this in the retina. Then Saad would do the retina experiments, and then he would tell Matthias, I predict this in perception, so go and do it in perception. And we went back and forth in a very, very enlightening collaboration that we learned a lot from. So how did it all start? So we started by going now to human perception, the output of the brain. And we will study saccadic suppression, so exactly the same phenomena. You make a saccade and then you flash stimuli. But what we did now was to say, okay, if we want to exercise visual mechanisms, let's have people make a saccade, like this arrow, across a textured background. And so this is like we do saccades in real life. If I look at a forest, I make a saccade across a textured image, not like in the lab with just a gray background, okay? And so we created an image with statistics that might be similar to what you might expect from natural images, low spatial frequencies and so on. And we also created images that are at the limit of natural scenes, like that have higher spatial frequency content, so much higher detail in the images, which is not so common in natural scenes, but basically the limit of visual processing capabilities, okay? And in both cases, what we did was simply just make a saccade and then have a perceptual detection, okay? So the experiment goes as follows. These circles that you see are not something that the subject saw, but they're placeholders to tell you where the eye was starting from. So you start from one of the corners and then you make a saccade towards the center. And then at some random time around the onset of the saccade, we make a very brief probe flash, like we said earlier, okay? And it's a luminance pedestal that appears in one of these 4 locations that are highlighted here. So you don't see these squares, but these are just the placeholders for the locations. And subjects simply have to say where they found the flash. Okay, so if there's suppression, they will be guessing. Okay, so indeed we get saccadic suppression. So again, on the y-axis, it's percent correct. And on the x-axis is the time of the flash from the onset of the saccade. So 0 is the onset of the saccade. And so you get strong saccadic suppression. This is with the coarse texture, these low spatial frequency textures. And now the key is, let's change the property of the image. So if there's a motor command, it should be, I'm making the saccade suppress regardless of the image, okay? But what we find is that your suppression is weaker and shorter lived if you are making a saccade across a fine texture. So we did other quantifications, and it's really a big difference between coarse and fine that is masked by the fact that here you go to floor, you just go to a floor effect, whereas with other quantifications, there's really a big difference. So it's not simply that, okay, there's a command that says suppress sensitivity regardless of what you're doing, but there's a visual component. So suppression is stronger and longer lasting with a low spatial frequency texture, okay? So this is the first hint that perceptual saccadic suppression depends on the background image statistics, which suggests that perceptual saccadic suppression is associated with a substantial visual component to it. It's not simply motor command that says reduce sensitivity, okay? Okay, so now I said we should go back to first principles. So where does this image dependence originate from? And as I said, we want to go all the way to the retina, okay? So what did we do? We now go to Sad, and Sad has mice that he basically dark adapts and then sacrifices, and then you isolate the retina, and then you cut it to flatten it, and then put it on a microelectrode array, okay? And then you project images to it, so it's as if it's seeing an image, okay? And the image that we presented is basically the same texture that the human subject saw, but we scaled it to the size of the eye of the mouse, because the eye of the mouse is much smaller than the eye of the human, but we scaled it in a matched way to the retinal ganglion cells, basically, okay? So the retina sees the image, okay? And then from the perspective of the retina, as I said, a saccade is an image translation. So I'm going to translate this texture here rapidly, okay? And that's a saccade from the perspective of the retina, okay? So now we look at an example retinal ganglion cell, and time 0 on the top here in the red bar is the onset of this saccade from the retinal perspective, okay? And what you immediately see is that a saccade from the retinal perspective is a stimulus. So it's not something that's suppressed. In fact, it's a stimulus, it's a motion, right? So there is a response by the retina to the saccade, okay? And then we do the exact perceptual experiment. So we present a brief flash, okay? In this particular case, the flash is indicated by this orange thing at 2 seconds. And then the retinal ganglion cell, of course, responds to the flash. Okay, so this is a sensitive neuron, and it has a response to the flash. So this is exactly the experiment that we did with perception, right? There was a flash, and then you measure the response to the flash in the retina. In perception, we just asked subjects what they saw. Okay, so as we did with perception, we changed the time of the flash relative to this saccadic event to the retina, okay? And so this retinal ganglion cell responds very briskly to the same flash, so very strong response. But what's interesting is when you come closer to the saccadic event. So here, if the flash is here, then the ganglion cell already returned to baseline, okay? But it's still impaired in its sensitivity to the very same flash that it saw later, okay? So there is a reduced sensitivity to the flash. And then if you move the flash even closer and closer to the saccadic event, you see that the neuron now is incapable of detecting this additional flash, okay? So if you plot the envelope, you see that there's basically, essentially, saccadic suppression in the retina. So the perceptual experiment that was done, if you present it to the retina, you get suppression already at the level of the retina. So the critical question is, does this suppression at the input to the visual system, does it actually also depend on these background textures that we tested with our human subjects? So is perception at the output of the brain consistent with what the retina does? So now we can take this retina, and then for these same retinal ganglion cells, we present the coarse texture and also the fine texture. Again, matched to the size of the eye of the mouse. So what do we find? We find something really remarkable, because both of them will have suppression, as you might expect. But now if you compare the strength and duration of suppression, you'll find that with coarse textures, the retinal ganglion cells are suppressed more. So if you compare here and here, the retinal ganglion cells are suppressed more with coarse than with fine textures. Exactly like perception at the very output of the brain. Okay? So you can put them next to each other now, and you see on the left is retinal suppression, and on the right it's perceptual suppression, okay? And so we see that for both the retina and for perception, there is a stronger suppression with a coarse texture and a longer lasting suppression in both cases. I'm going to come back to we can come back to pre-saccadic stuff later if you wish, okay? So this was very robust. We tried it in different species, mouse, pig, and now in more recent work also with we got access to non-human primate retina. and we got the same effect basically, that there is suppression of visual sensitivity with these flashes after the saccadic event for the retina, and the dependency of the suppression on the image across which you make the saccade is basically the same as in perception at the very output of the brain. And the fact that it happens with multiple species suggests that it's basically a general phenomenon of visual processing and not really specific to the mouse or to the monkey or whatever. And we also see it in humans in terms of perception. Okay, so this is very interesting, but it begs the question of, okay, what is it about the motor commands now? Because we know that a structure like the superior colliculus will generate a motor command for sure. We saw it in Shadlin's talk yesterday, and we know that the colliculus is connected to cortical visual areas, so there is information about the visual signal. So what is the motor command doing now? Okay, if it's all done in the retina, okay? So the hint comes when you compare the time course of suppression in the retina to the time course of suppression in perception, okay? You see that in the retina, suppression is very long lasting. You know, by 600 milliseconds or more, the sensitivity of the retina is still impaired. Whereas in perception with real saccades, you recovered by 100 milliseconds. So there is much faster recovery in perception with real saccades than with these image shifts on the retina, okay? So this might suggest to us that what your motor command is doing is actually not what people have suggested, which is to send a suppressive command to the visual system, but in fact the motor command might be enhancing sensitivity in order to basically shorten or compensate for the long-lasting suppression that originates visually from retinal mechanisms. So this is almost the exact opposite of what the classic idea of a motor command doing to saccadic suppression is. Okay, so can we see evidence for this? Well, we can now take the retinal predictions and say, I can now go back to the human lab and then I can run the perceptual experiment with humans, but now without any saccade. So I'll just have people fixate and I will translate the texture exactly like we did to the retina, okay, without any saccade. So we get rid of the motor command. So from the retinal experiments, we have several predictions that we should expect, okay? So prediction number one is that we should still get strong perceptual suppression with an image translation because the retina shows it, okay? So indeed, that's exactly what we get. So we, at time 0 is the shift of the image, no saccade command, just the shift of the image. And then we present flashes at different times and we get strong suppression. We can discuss the pre-translation effect, but we still get a pre-translation effect. So we think we can understand it. The second prediction is that since the retina depends on the image statistics, then perception should also depend on image statistics without a motor command. And that's exactly what we find. With a fine texture translation, you basically get weaker and shorter-lived suppression than with a coarse texture translation, exactly like with real saccades, exactly like with the retina in an ex vivo retina preparation. And most importantly, the third prediction is that now if you get rid of the motor command, you should expect longer-lasting saccadic suppression than with real saccades. And again, with the very same subjects, we test them twice, once with real saccades in this orange curve, and once with these retinal image translations of the shift. So we just simulate the saccade but get rid of the motor command, and we get longer lasting suppression, basically, as might be expected from the retina. So this suggests that the motor command, instead of sending a signal to suppress sensitivity, might actually be trying to sort of cover the basis after the retina has created this longer lasting suppression. Okay, so visual retinal mechanisms can explain properties of perceptual saccadic suppression, the image dependence, and motor-related mechanisms are now kind of perceived to do something the opposite of what was originally suggested, which is to try to enhance sensitivity, to try to compensate for the long-lasting visual effects. So perceptual saccadic suppression, in our view, starts already in the retina. Okay. Now, this can now take us all the way back to the beginning of my talk. So now we have this fresh perspective on visual mechanisms being important for saccadic suppression. So now we thought, well, okay, we started with this sort of defeat with respect to selective saccadic suppression because we still don't quite understand it despite the Superior Curriculus results. Okay, so now can we revisit this selective property of perceptual saccadic suppression from the perspective of visual mechanisms? And we can ask ourselves, is this phenomenon fundamentally a purely visual phenomenon? Okay, so this might seem heretical with respect to the original Nature paper and how it was framed, but maybe with this insight that we got from the retina, we can try something, right? Doesn't hurt to try. So that's exactly what we tried. We tried to do their experiment exactly like what they did. There's a saccade, so you start from a spot here and you make a saccade, and then at some point we flash a low or high spatial frequency grating. But the difference that we introduced was that now around the monitor, we created a context, a peripheral visual context. In this case, it was just a coarse texture, okay? But otherwise, it's the same experiment. You make a saccade with a flash, okay? And when we did this, we basically replicated the results exactly. Like this is exactly what I showed you earlier on the left. And on the right, we just take the difference between the two curves to highlight that, okay, the low spatial frequencies have strong suppression and the high spatial frequencies do not have strong suppression. So this is exactly their experiment. We replicated it. Okay. Now, we asked ourselves, what is this experiment doing from the perspective of visual properties, from the perspective of the retina? If I'm making a saccade in this direction, then what was the image that was on my retina before and after the saccade? Okay, so take a purely visual perspective on this phenomenon. And if the saccade was up to the left like this, then the image on the retina, like if this is the center of the retina here, the white dot, then the image on the retina was basically the monitor up on the left, and then it gets translated down to the right by the saccade, and then you are at the end of the saccade, and then there's a flash. Okay, so from the visual perspective, that's what the saccade experiment did. So we ran the same experiment now with fixation, so without a saccade, but with this visual perspective of a shift of the visual monitor and the flash. And to our surprise, we got exactly the same phenomenon that they showed 25 years ago in their Nature paper, which is that low spatial frequencies are suppressed without any saccade, and high spatial frequencies are not suppressed. So you can get selective suppression of visual sensitivity without any saccades at all, just with an image shift, okay? And therefore, with or without a saccade, you can get selective saccadic suppression, basically exactly what they found, just based on pure visual processing, okay? So now we went even more crazy and we said, well, okay, if it's pure visual processing, let's now take this monitor, we keep the monitor as it is, but we change the surrounding context. So just the surround, okay? And we just change the context from the coarse to a fine texture. Otherwise, the experiment is exactly the same. And now we run the experiment without any saccades, just a translation, but just with a different context. And we just basically do the simulated saccade version. It's the exact same thing, but we just change the surrounding area around the monitor. And what we find, surprisingly, is that there is non-selective suppression now. So the curves are parallel to each other, and this difference measure is constant. So basically, low spatial frequencies are suppressed similarly to high spatial frequencies. Okay? So this made us go crazy, right? Because now you just did an image shift, and you got rid of selective suppression. This was no saccade. So we said, well, there's a strong prediction here, because now we can predict that the selective property of saccadic suppression for low spatial frequencies in this paper can actually be violated, like our Superior Curriculus neurons violated it, with real saccades if we simply change the visual surround. And that's exactly what we found. So we repeated the exact same experiment, but we just changed the surround around the monitor from coarse to fine, and now with real saccades, perisaccadic flashes, we still get non-selective saccadic suppression. So we violated this Nature 1994 paper, just like our superior colliculus neurons violated it. So basically, simply changing the visual context is enough to eliminate selective saccadic suppression of low spatial frequencies. So selective perisaccadic suppression of low spatial frequencies, in our opinion, is a fundamentally visual phenomenon. And this is interesting because, as I said, in the colliculus, we found neurons that match the phenomenon, are selective, but we found neurons in the same structure that were non-selective, and they violated this phenomenon. And this is interesting, actually, because if you think about it, the deeper layers and the superficial layers have different patterns of lateral interactions and surround effects from each other. And so now we might have a handle on understanding why we got so different violations of the phenomenon in the superficial layers when compared to the deeper layers. And obviously this is creating a lot of new questions that we're trying very hard to answer right now. And so these are basically our current steps at trying to unravel the neural mechanisms for perceptual saccadic suppression. And when we first posted some of the results on bioRxiv, someone tweeted a sentence that I like because it reflects what I also feel. So he wrote, "This changed my view on what causes saccadic suppression and what is the contribution of the movement command. It's a good feeling to be persuaded by experiment, and I fully agree with this assessment." So with that thought, I will thank you for your attention, and thank you again for the very nice invitation to join you.
Thank you very much, Ziad, for the very nice presentation. So the floor is open for questions. We have about 5 to 10 minutes. If there's any questions, okay, Dr. Tagizadeh, please go ahead.
Hi, can you hear me?
Yes.
Thank you very much for the very nice presentation. So my question is about the pre-saccadic motor commands, basically. So if you so you showed us that motor commands are correcting for the suppression. That was what the data was showing. Now, if you make the target, the saccade target and the saccade onset time 100% predictable for the system, does that affect the level of correction that the motor command has on the suppression?
That's an interesting question. So we can make the timing of the instruction to make the saccade 100% predictable, but the saccade itself is still variable, in reaction time, basically. So, unless you make back and forth, so if you just keep going back and forth, then you synchronize with the frequency of the jump, and then the reaction time becomes a bit more deterministic. But if you want to avoid this back and forth stuff, it's hard to have deterministic onset time of the saccade. But our hypothesis, if we do it back and forth like this, our hypothesis is that if you are able to modulate the saccade command somehow, you should change the properties of the perceptual suppression. Yes.
Thank you.
Any other questions? So let me ask a quick question to Ziad. So two quick questions, and I want to have your thoughts on it. First of all, the saccadic suppression, as you showed it, and many people have shown, starts before saccade, the start of the suppression. So there must be a motor component for that, because at that point we don't have the movement on the retina. One question, and the second question is that in one of the slides, unfortunately, they didn't have numbers, so I don't remember which one was it. Your psychophysics data, when you moved the stimulus for the subject, still the duration was different from, was still longer than the actual saccadic suppression. So how do you reconcile this one? So the first one is the starts before saccade, second is still physics is longer. So what's your thought on that?
Yes, I can show the slide because I think it basically, so both of them I think can be answered here. So the pre-saccadic suppression is something that we also see, like as you see from this red curve, we also see without the saccade. So if we start the translation of the image, then we flash the still before the translation, okay? Then people are still impaired at seeing it, okay? And I think, and similarly with real saccades. So this is what people in psychophysics might call backwards masking, basically. So the problem with perception is that perception makes the report a few hundred milliseconds after everything has been done. So the stimulus, the flash comes on, then the background translates, and then a few hundred milliseconds later, the subject says, I saw it or not. And so there is a misattribution, I think, in backwards masking in the perceptual report. So using pre-saccadic suppression in perception as a means to say that there must be a motor command is not very robust, because you get pre-saccadic suppression even without saccades. It's just backwards visual masking, and I think what happens in the visual system is that there's a credit label misalignment, basically, that happens. So what one needs for pre-saccadic stuff is neural activity, to do it neuronally, and before saccades there's a motor command that has pre-saccadic elevation and so on, and there it can still happen, okay? But we can't use this perceptual evidence to argue that this is evidence of a motor command. It's just backward masking. Now, for the second question with why is the suppression longer in the red curve than in the orange curve? So that's a nice question. We don't know. We just predicted it from the retina, because in the retina, when we measured suppressed sensitivity in the retina, we found that the suppression of the sensitivity of the retinal ganglion cells lasted for so long, and we were confused because perceptual suppression with saccades is very short. So that's why we did this experiment. We said, well, if it's the retina, then maybe if we just translate the image without the saccade, then the suppression should also last for a long time. So why it's happening, or how is the saccade command compensating, is a big open question. And that's what we're trying to solve now.
And one final thought: what is your take on the words in summer, inactivation? So they clearly show if you inactivate. So there must be a corollary discharge from superior colliculus going in and with mucimal. So how do you then reconcile these two findings? Your finding that is mainly visual based versus that the evidence for
exactly. Yeah, so I go back to this slide here, because basically this slide, Sommer and Wirtz have identified a corollary discharge pathway from the deeper layers to the frontal cortex. Okay, so it was basically from the deeper layers to the medial dorsal thalamus to the frontal cortex. And then Berman and Wirtz later looked at, the pathway from the superficial layer to MT via the pulvinar, and they looked from superficial to MT. So Sommer and Wirtz studied this pathway of deeper to frontal cortex from the perspective of mislocalization, so not really saccadic suppression, but another perisaccadic phenomenon, whereas Berman and Wirtz looked at superficial to cortex for saccadic suppression. So they looked at only from SGS to cortex. And there they argued that this is the pathway for saccadic suppression, but what they didn't look at is the deeper layers. And we find much stronger saccadic suppression to the deeper layers. So we think that saccadic suppression goes through the Sommer pathway, not through the Berman pathway. That's our hypothesis.
Good. And I will read one question in the chats for you. Alessandro Ziad says, thanks for the great talk. Is there any evidence for saccadic suppression for higher visual areas like IT, FBF, and DLPFC? And I think you kind of answered in your previous answer, but maybe you want to elaborate again one more time. Saccadic suppression on higher areas, IT, FBF, DLPFC. The FBF, DLPFC is the Sommer and Wirtz, of course, but what about IT?
Yeah.
Right.
So I would suspect that even IT would show it basically. So there's I think Michael Paradiso has some work on V1 with these probe flashes, and the V1 shows it, and FEF also shows it, and I would suspect that ITS would also show it. Yes, yeah.
Very good. Any other questions? We have time for one more question, maybe. Dr. Sanai, please.
Hi, Ziad. Thanks for the great talk. I was thinking about the data that Alex Thiele has with Hoffman in MT, and if they see psychotic suppression in MT, and they showed some probably you know that trials monkey make a saccade, and some trials they just show a translation of the stimuli, and they clearly show that psychotic suppression only happens when the monkey makes a saccade, not any mistranslation. What do you think of? Do you think any discrepancy between your data and theirs, or?
Yeah, so this is a very good point, actually, and I think this is a point that needs to be clarified in the literature. So what they looked at is basically the response to the image translation in MT by saccades or without saccades, basically. And this is what people in perception call saccadic omission, like just basically the disappearance of the image shift. And people in perception, in addition to that, they added these brief visual probes to catch the sensitivity, the instantaneous state of sensitivity of the visual system. And that's, I think, where the problem lies. The problem lies in this probe approach to the experiment. So Thiele and Hoffman looked at how is the translation canceled, and the people in perception call it perceptual saccadic omission. But the stuff that I talked about is, okay, look at what happens when you probe sensitivity with a flash. And that's a problem. Sometimes the literature mixes the language between the two, okay? And I think we need to clarify it, because I think the problem with the approach that we looked at here, which is brief flashes, is a stimulus-stimulus paradigm. For the retina, it's like a translation with a flash. And so that's a stimulus-stimulus paradigm. And I understand why perceptually you might want to do this as a stop motion photography or whatever, but mechanistically it might not be the best way to go. And I think it might be best to take the Thiele and Hoffman approach in the future to look at these intrinsic responses to the saccade shift itself. And this is something that needs to be done, yeah. But perceptually, people call them two different things: saccadic omission and saccadic suppression.
Thank you.
Okay, thank you very much. I think it's time now